Okay, take out your yellow notes. Mitochondria. The mitochondria are little organelles that release energy for the cell to use. This is where cellular respiration takes place. They call this the powerhouse of the cell because nutrients go in, as well as oxygen, and energy comes out. Organelles called the endoplasmic reticulum. They move the substances throughout the cell. So, these little things are mostly located outside the nucleus because the true job of the cell is to create copies of the chromosomes that are inside the nucleus so they can reproduce. So these little apparatus called the endoplasmic reticulum move things throughout the cell. Golgi apparatus. The Golgi bodies, or the Golgi apparatus, are the packaging and distributing centers of the cell. It's kind of like the post office. They package things up. They put things together for the cell. These little organelles are like closets, or they're like places where things are stored. So we call these the vacuoles. They store food, they store water, and they store waste in the cell. Plant cells have big vacuoles, where animal cells have many vacuoles, but little ones, tinier ones. The lysosomes, they're only found in animal cells. Their job is to digest the nutrients and break down old cell parts. So lysosomes reminds me of like Lysol. They break things down. These little organelles are called ribosomes. They make proteins that are needed for growth. So this part is really important because they help build up the DNA. The animal cell. The animal cell is generally round in nature. It has only a cell membrane, whereas the plant cell has a cell wall and a nucleus in the middle. It has all different parts that help the cell do its job. These organelles are only found in plant cells. They're called chloroplasts. Chlorophylls the chloroplast. This green stuff inside is a pigment called chlorophyll. This is what the plant uses to make its food. Plants are generally green because of the chloroplasts. Now, other cells also have chloroplasts, like the Monera have chloroplasts, and so do some protists have chloroplasts. This is what makes the cell an autotroph. So the green pigment helps absorb the sunlight so that the plant or protist or Monera can undergo photosynthesis. Photosynthesis is the life process that allows plants to make their own food. The rigid outer part of the cell is called the cell wall. This is only found in plants. It's made up of something called cellulose. It gives the plant its support. The cellulose is like a starchy, sugary material that coats the outside. So plants have cell walls and cell membranes. Only animal cells have cell membranes, but the plant cell has that outer structure so that it gives it support. The plant cell is square in nature because of the rigid cellulose. That cell wall makes it really strong. So you think of a house. A house is made up of walls. That gives it that structure, the rigid shape. Okay, so on this part, we're going to put together things that are similar and things that are not so similar in the animal and the plant cell. So the first thing we're going to look at is the nucleus, the cell membrane, the cytoplasm, the mitochondria, the ER, the Golgi bodies, the ribosomes, and the fact that both animal cells and plant cells all have to undergo the life processes. So all these things in the middle are what the animal cell and the plant cell have in common. However, the animal cell has something called lysosomes, where the plant cell does not. It doesn't need them. Because the plant cell has large vacuoles, it doesn't need lysosomes, because the vacuoles pretty much do the same thing as the lysosomes do. The animal cell has small vacuoles, so it needs those lysosomes also to break down parts. The plant cell has chloroplast. This is where the food making comes into place. The plant cell also has chlorophyll. The chlorophylls the chloroplasts, so they're one and the same because the chlorophyll is inside of the chloroplasts. The plant cell also has a cell wall, which the animal cell does not have. The animal cell has tiny vacuoles. They have many small vacuoles. The plant cell has one large vacuole. So the basic differences that the plant cell and the animal cell have are the structures, the chloroplast, the chlorophyll, the cell wall, and one large vacuole. The shape is also a difference. The animal cell is generally round in shape, where the plant cell is more rigid. It has more like a rectangular or square shape. So you could see that the one on the left is definitely the plant cell. You see the rigid structure, you see the cell wall, you can see the chloroplast, whereas the rounded structure is more the animal cell, because it doesn't have that plant cell wall to make it rigid. So it can have any shape it wants, and it generally tends to be like a rounded shape. Okay, all cells need to allow materials to come in and out. And where did that happen again? When we regulate the materials coming in and out, that happens in the cell membrane. So both of the cells have to allow materials to come in and out. So diffusion is that process that allows materials to move in and out. But diffusion happens without any energy. So the plant and the animal cell both want to reach equilibrium. They want things to be equal. So if something is missing, it's going to allow it to come through so that everything on the outside is equal to whatever's on the inside. So certain structures, certain things easily diffuse, like water. If the plant cell doesn't have enough water or the animal cell doesn't have enough water, it's going to allow water to diffuse through. Passive transport is diffusion. This means it doesn't happen with the use of energy. We don't need any energy. It just happens because it wants to release, it wants to maintain its equilibrium, so it allows things to come through. Active transport, it needs energy. So if the plant or the animal cell needs food, it has to actively allow it to come through. All right, osmosis. This is the movement of water through the membrane. This is a form of diffusion because it doesn't require any energy. So if there's not enough water, it's going to allow it to diffuse through. It's also going to allow sugar to diffuse through. So they want everything to be equal, so the water has to go through. That's what osmosis means, allowing water to diffuse through the membrane. The reason why we say that viruses are not living is because they don't have that cell structure. They're not considered living. Now, does that mean that they're always going to be considered non-living? No, it just means right now they don't really have all the characteristics of life. So a lot of people have controversy over this. A lot of people don't believe that viruses are non-living. They think they are, but they don't have all the characteristics of life. So if we follow that rule, viruses cannot be considered living. All this information was developed and discovered basically because of the microscope, because you really can't see cells with your naked eye. The naked eye means you don't have an aid of an instrument. When people say that, the naked eye, that just means your eye without anything on it. So if you have to use glasses, you have an instrument to help you see. So the microscope was an instrument that was developed in the late 1500s that helped us see the cell and what was inside of it and what it was made of. Today we have lots of different types of microscopes. The microscope you're going to use in the classroom is called a compound microscope. Just like a compound word has two parts, our microscope is a compound microscope because it also has two parts to allow us to see the image. We have an ocular or an eyepiece, and we have an objective lens. Those two lenses work together to help us see. So the compound microscope is what we're going to use in the class. The compound microscope has two lenses, like I said before, the eyepiece and the objective lens. So both of those pieces allow us to enlarge the image. So when we talk about magnification, that means how large is the image under the microscope. So if we're looking under the microscope and you put your eye on the top piece, and that top piece is right here, this is the eyepiece. The objective lenses are right here. They both have a piece of glass in them, a curved piece of glass, like a lens, that allows you to see the image bigger. Not much bigger, though. Our microscopes can probably just show you an image 400 times bigger. Whereas if we use an electron microscope, it can show you things thousands of times bigger. Our microscope is called the compound microscope. So if you use the microscope under medium power, you're using the middle objective lens. And you will magnify with the eyepiece and the objective lens. So if the eyepiece has a magnification of 10 and the objective lens has a magnification of 25, you multiply them together and you get total magnification of 250. Our microscope has many different parts, and we're going to use these parts in the classroom. And you're going to tell me what they do. So right now, and when I ask you to pick up the microscope, you need to make sure you pick it up with the arm and the base. You're looking at the eyepiece, and this is where you put your eye. And a lot of times, people that have long eyelashes kind of like tickles them, and it's hard to see. Sometimes people like to close one eye while they're looking through it, and you physically have to close it by putting your hand over the other eye. These objective lenses can move. They move around by a use of this piece we call the nose piece. So you have the objective lenses, and when you want to put the slide underneath, you use these stage clips. The slide will go on something called the stage, and you see this little circle that allows light to go through, because you need to use the light so you can see the image a lot crisper. So there's two different knobs, and the knobs in the microscope we're going to use may have both the knobs together, but then they also have, some of the microscopes we have, the knobs are separate. So the coarse focus knob or the coarse adjustment knob is the bigger knob, and the fine adjustment knob is the smaller one. So there are specific rules on how to use these knobs, and we'll talk about that in class. We'll talk about what the diaphragm does as well. So we're going to stop right now, and then you're going to complete whatever you need to complete according to the directions in the smartboard. Okay? Good job.